My name is Jurczyszak, and I am a candidate for a Bachelor of Science degree in biology. My CaNCURE project was on the localized sustained delivery of cancer therapeutics via biodegradable PLGA implants. Poly-ADP ribose polymerase, or PARP, inhibitors induce DNA damage by preventing single-stranded break repair within the base excision repair pathway. During DNA replication, single-stranded breaks become double-stranded breaks. PARP inhibitors induce synthetic lethality in cancer cells with BRCA1 or 2 mutations, which are deficient in homologous recombination, which is a double-stranded break repair method, and therefore causes BRCA-deficient cells to lose the ability to repair themselves. As DNA damage in cancer cells treated by PARP inhibitors accumulate, they undergo cell cycle arrest and eventual cell death. As part of my co-op, I learned how to fabricate polylactic coglycolic acid, or PLGA, implants that contain PARP inhibitors. Conventional drugs are typically administered intravenously or orally, with only a fraction of the drug molecules accumulating at the site of the tumor. PLGA implants, however, degrade slowly over time and can be implanted at the site of the tumor, allowing for sustained delivery and localized delivery of cancer drugs. This minimizes off-target effects, improves therapeutic efficacy, and allows for lower dosing of the drug, thereby reducing toxicity. I learned about two different methods to manufacture a drug-loaded PLGA implants: hot melt extrusion and a solvent-based method. For hot melt extrusion, a mixture of PLGA and the drug is made, with the ratio depending on the desired concentration of the drug. After mixing the solution, the powder is transferred into a syringe, which can result in the loss of some material. The syringe is then attached to silicone tubing, and the syringe containing the powder is heated in order to create a viscous liquid, which is extruded into the tubing. After the liquid is dry, the implants can be removed from the tubing and cut to the desired length using razor blades. Solvent-based implant fabrication takes advantage of the solubility of the drug in order to produce an aqueous solution of the drug, which is then dried. Chloroform is added to PLGA to dissolve it, then the desired drug is added to the PLGA solution. DMSO is added to dissolve the drug, and sonification and centrifugation aid the dissolving process. A syringe is used to collect the aqueous solution, and tubing is attached to the syringe. A syringe pump is used in order to extrude the solution into the tubing. Then the solvents are evaporated by placing the tubing into an oven overnight. I learned how to perform cell culture work using the BPPNM cell line, which was obtained from the Weinberg lab at MIT and the Broad Institute. This cell line has loss-of-function mutations in the BRCA, P53, P10, and NF1 genes, and overexpresses the MYC oncogene. These mutations are characteristic of high-grade serous tubo ovarian cancer cells. I was able to learn many different cell culture skills and techniques, including cell passaging, cell counting, and performing MTS assays. Part of my co-op work was developing an in vivo model of BRCA-deficient ovarian cancer using BPPNM cells. The BPPNM cell line is a murine cell line, so immunocompetent mice were able to be used, which is significant as this model could be used in the future for testing immunotherapies. The studies we did included 5 immunocompetent female mice that were placed into 2 experimental groups. 3 mice were in the treatment group and received an implant containing TLZ, while 2 mice in the control group were given a blank implant. The mice were inoculated with 5 million BPPNM cells. 3 weeks later, the implants were administered to the mice via an intraperitoneal injection using an 18-gauge needle, which is used in the clinic. I found my co-op to be an extremely meaningful experience in which I grew not only as a scientist, but as a person as well. I learned a wide variety of lab skills, including drug formulations, cell culture work, and working with animal models, and learned skills such as restraint and intraperitoneal injections. I also learned about the relationship between research and the clinic, the importance of translating research from the bench to the bedside. In the future, I hope to pursue a career in medicine, specifically in oncology and women's health, and contribute to research in order to improve patient outcomes. Thank you.